easily recognize some of our tissues today because I do not recall all of you from the lecture hour, so it would be beneficial if you all attend the courses, in my opinion, all right? Not only for my lesson, but for the next section as well. So, okay, more close. Today we will see the digestive system tissues, digestive system tissue sections obtained from some organs. And during the lesson, to remind you, we were starting from the mouth. The upper part of the digestive system was starting with the mouth, and then continuing with the esophagus, and then we will end up with the stomach today. Okay. So the first part of the mouth and the first part organ in our mouth, we will show you the tongue. The tongue in our mouth is the first organ, let's say, in the digestive system. And as you see, the tongue is very well identified with the, okay, once more here. Can you hear now? Is it okay? As you see, the tongue is lined with an epithelium. Since I'm sure the ones who attended the course easily recall, we are always investigating by the layers. In each organ, we are investigating the layers of the organ. So starting with the mucosal surface, the tip of the tongue is lined with a stratified squamous epithelium. Here you can easily recognize the stratified squamous epithelium with many cellular layers, and the squamous cells on the uppermost layer. Okay. Uppermost layer is squamous cells, but stratified, so there are many cells resting on each other. And then we have a thin lamina propria, and then we have the submucosa filled with the blood vessels and other elements. And then, as you see, we are reaching to the muscularis layer here, as you see, trying to get to the muscularis layer. Okay, here the muscularis layer, and there are some other glandular structures invading between those muscle bundles, let's say. So this is the tongue, and it is the tip of the tongue. So at the tip, it is a uniform layer of stratified squamous epithelium. And the tongue, as we go through the upper mucosa, I am trying to find some differences through the tongue. As you see, it is still stratified, and sometimes the lamina propria and the submucosa are getting some invasions through the epithelia, and those invasions should be more clear when we get to the papilla structures. At the moment, more about the stratified epithelium, and then here is our first papilla structure. So, how we recognize this papilla structure? We can easily recognize the papilla structure is the resting still over the mucosal surface. But as I told you, the lamina propria and the submucosa is making invasions through the epithelia. But still, we have a continuous basal lamina of the epithelia. So this basal lamina is clearly seen with the bluish, greenish color belonging to this preparation, histological preparation. So here the foliate type of papilla is clearly identified with the lateral sulcus, lateral sulci, okay, one on this side, on the left, and another on the right. And this sulcus are also lined with the special type of epithelia. You should remember from last year, neuroepithelium type here are resting, which we call the taste buds. Okay, that's why our first preparation is named as the taste buds. Here is the taste buds. They are resting in the sulci. Okay, let me have a larger magnification. Next magnification with some light adjustment. Here is the taste buds. Okay. They are really, those taste buds are really oval shape of, let's say, cell gatherings, but they have special neuroepithelium type of cells functioning for the taste recognition. Okay. Still, we are observing the stratified squamous epithelium, but the cells in this stratified epithelium, we have the spatial taste buds, and that's why the foliate papilla is very common with its rectangular shape. Okay, the rectangular shape giving us a tip, and then two sulci, and then the base, the lamina propria, giving us a rectangular shape. Okay. Now, going back to the lower magnification, and we can still observe those other tissues, other cellular elements. Still, we need to. Okay. We can observe the lamina propria, the submucosa filled with the glandular structures. You remember we have some lingual and sublingual glands. They are still also resting in the tongue itself. And then, more going deeper, we are coming to the muscle layer, muscularis externa or tunica muscularis. You can understand that those muscle cells are the striated type of muscles, so the tongue, the organ itself is very gives the motility function of the organ. Okay, and there are other small sections of blood vessels, blood capillaries, or if you go deeper, still you can observe some glandular structures. Here there are some glands. And then I'm going deeper, a thick section of tongue. So still observing some blood vessels. And then maybe another blood vessel here. Okay. And our section is cut through the basal surface. So not this is not a gross anatomical section. So the basal layer is cut through, and that's why we do not observe any epithelia here. But here is a big nerve section, and other capillaries or vessels are identified. Okay, our main interest will be at the mucosa, the mucosal surface, and then the layers, mucosa, lamina propria, submucosa, and then tunica muscularis. Alright. I'm going to the next section, which will be the esophagus. And then lastly, tooth, okay. When we investigate the esophagus, you can always have a compound structure or compound section, both going through the trachea anteriorly and the esophagus posteriorly. Okay, so if you have your section and put it under the microscope, please first of all try to recognize the structures at the lowest magnification. So the esophagus lumen is clearly identified, okay, by the dentations. Let's say some imaginations, and the trachea lumen is very circular and is very straight lumen. So we are now interested in the esophagus lumen here. So I will also try to introduce you the layers of esophagus. So it is the same for all digestive tract. As you remember, we can still observe the mucosa, surface, and then the next layer with the lamina propria and submucosa, and then the tunica muscularis, tunica externa, with the lowest magnification, and then lastly the adventitia, or some parts would be the serosa. Going backwards, still adventitia, tunica muscularis, submucosa, small bundles of still smooth muscle bundles here, muscularis mucosa, okay, muscularis mucosa, and also including the lamina propria, means that we are in the mucosa, and then the lumen inside. Once I make a bigger magnification, I can observe that the mucosa of the esophagus is very similar to the mouth and the tongue that we were observing in the previous one. So similarly, it is also lined with a stratified squamous epithelium. So as you see, the uppermost are the squamous, and many cells resting on each other, stratified, and the ones at the, okay, slowly, at the base, more round nuclei are the basal cells, but the ones on the lumen facing cells are the squamous cells. Okay, and then the next layer would be the lamina propria. Here the lamina propria includes more the connective tissue, and since our section is stained with mallorazan here, as you see, the connective tissue fibers are stained, excuse me, connective tissue fibers are stained in blue color, but our nuclei are stained in red color. So you can easily understand that this section is stained with mallorazan staining, and through the lamina propria, the bundles, individual bundles of muscle cells are belonging to the muscularis mucosa. So they are also the smooth muscle type of cells. And neighboring to those bundles, you can still observe some blood capillaries or blood vessels, and there might be some other neuronal endings as well. So going back through the submucosa, it is a broad area of the submucosa with okay, better, with many fibers. So many fibrous sheets, collagen bundles or elastic And then the next layer would be the tunica muscularis or tunica muscularis externa. Here, depending on the part of the esophagus which our section is obtained, we can sometimes observe some straightened muscles, or some of the part we can see the smooth muscle cells. I'm sure the ones in the lecture would remember that upper 1/3 of the esophagus more straightened, lower, middle 1/3 portion would be a mixture of straightened and smooth muscle, and the lowest part would be completely smooth muscle. Okay, since that's why maybe we can still observe some situations if I make some adjustments, but here at the moment not very much visible. But I'm sure if you go through your preparations on your microscope, you can see some different muscle types. Okay, and the last layer was the adventitia, the outermost layer, the adventitia, and adventitial elements are clearly visible. Okay, our next section is the stomach. Here is a section taken from stomach, let's say longitudinally, and I can observe the surface on the right side, on the tip of the arrow, and I can observe the serosa and the other tissue elements on the left. So again, starting with the lowest magnification, first of all, you should identify the luminal side, okay? The luminal side with the mucosal gastric pits and gastric mucosa, but the outer surface would be the serosa, okay? So first of all, it's easy to recognize the mucosa, the lumen of the stomach, and we go through the lumen, and it is still some showing us the pits, gastric pits and gastric glands, and then okay. Now, first of all, we identified the mucosa, and then through this low magnification, we can still observe the other layers. Mucosa belongs until to the includes the lamina propria and extends until to the submucosa. Here, the connective tissue elements, as you see at the tip of the arrow, the submucosa, and then the muscularis externa, having a broad, thick layer, and you can remember that the tunica muscularis here, muscularis externa, has 3 layers, circular, longitudinal, and oblique sections as well. So that's why a thick section of muscle layer, all going around the stomach tissue. Now we can increase our magnification to see more about the types of the cells. First of all, the mucosa is lined with the columnar epithelium. Okay. Now, different from esophagus, the surface of the stomach is lined with a columnar type of epithelium. How do we recognize it? Here is the columnar type of epithelium having the spindle-like, spindle-shaped nuclei and perpendicular cytoplasm, rectangular cytoplasm, and that's why, as you see, lining all through the surface is belonging to this columnar epithelium. But this epithelium covers the mucosal surface and continues with the gastric pits. That's why, as you see, it makes some imaginations through the stomach surface. So we call those imaginations as the gastric pits, still lining with the columnar epithelium. And another type of cells are lining this part. We say the mucous type neck cells. Okay, the tips, the surface tips are lined with the columnar epithelium, and then surface mucous cells. And then we will go more in deeper to the glands now. Let's say here. Everybody hearing me? Listening? Okay. Thank you. Now, yes, here we are going through the glands. As I told you, until now it was the surface epithelium, columnar epithelium, and some mucous neck cells, and then we are more in the glandular structure. So since this is still continuous with the gastric pits, it is directly continuous, and they secrete their material to the stomach surface. And the cells resting here are identified belonging to their staining characteristics. As you see, some of the cells are stained more eosinophilically, okay, more pinkish or reddish cytoplasms. They are very well characteristics of the parietal cells. Okay, those parietal cells here I'm pointing are more pinkish, reddish cells. But some other cells, more basophilic staining cells, are also present. When we go deeper through the glands, they are the next cells, which we call chief cells. Okay, the chief cells are more basophilic, but the parietal cells are more eosinophilic staining. And then the mucosa is end up with the lamina propria all going through this part, and then the lamina propria, okay, is the next. We can go to the next layer, which is submucosa. Here, the submucosa is still having some blood vessels and some neuronal sections as well. You can remember that those neuronal endings are called here in the submucosa are the Meissner plexuses. But what about the next level? The tunica muscularis, muscularis externa. As you see, the smooth muscle, and it is very, very thick. I will make a lower magnification. That's why there are many oblique orientation, transverse orientation of muscle cells, but usually the ones, the outermost ones are longitudinal and inner circular, and some oblique sections. And between those layers, we can still see some capillaries, and we can observe some another neuronal elements here, as you see. Okay. So you can recognize that this group of cells is staining, and having different shapes of cytoplasm compared to the smooth muscle cells. Here is the group of the mesenteric plexus. Okay, those cells are the mesenteric plexus ganglionic cells. So they have the neuronal input to the tunica muscularis externa. So we can see some other sections still, all while going through the stomach tissue. And the smooth muscle cells, as I told you, some of the smooth muscle cells have the spindle shape, some of them because of the orientation, some are showing longitudinal sections, or some cross sections with pointed nuclei, but they are all still the smooth muscle cell characters. Okay. And one more identity along this stomach. Let's say here you can see that we have the pits, and then, as I told you, and then the glands. But I want to go through this stomach section along more further, and then, as you see, the gastric pits are getting longer, but on the other hand, glands are getting shorter. So that shows me that the first part that we were investigating was the cardiac or corpus part of the stomach, but the next part would be more about the pyloric section, okay? So that those pits are longer and the glands are shorter, let me say. And also you can always see some lymphatic nodules. You remember in the lamina propria and the submucosa, there would be the gastrointestinal system protective elements of lymphatic nodules are present. So that's why some gatherings of lymphocytes present all over there. And then still about the pyloric section. Okay, there would be probably duodenum or intestinal junction over here, but our section was just cut through the stomach portion, so we do not observe any duodenum or intestinal portions. Okay, our last section is about the tooth. Just to remind you that the tooth itself is the is formed of the hardest calcified material present in our mouth. And it is they serve for the breakdown of food material into pieces and getting ready for the digestive processes. As you see, we can observe the crown part of the tooth, and going through this crown we can go through the neck portion. Here actually this tooth is individual tooth, not resting in the bony alveolar material, not in the mandible or maxilla, but this is individual tooth, so maybe it is difficult to recognize the neck portion. But we can understand that going further down, we are going more to the edges. And then the pulp section, we are going through the pulpa section. And the root section is more obvious here. And all through this root section, the invading neurons, invading, let me say, axonal extensions of those neurons are invading through this root section, and there are many blood vessel sections as well, all going through these two roots. And the lobes part. And another, if we make another magnification, we can observe from the crown part the enamel layer, mainly formed by the rods of animals, but the dentin material, more let's say alveolar type of cells here are resembling the dentin part. Okay, the enamel outside and the dentin inside. So I think that's all with the sections. Please do not hesitate to ask questions, and good luck. Thank you.